Lauren Bush has first-hand experience with food poisoning. Over a decade ago, she got extremely sick after eating a spinach salad. She suffered mysterious flu-like symptoms for 6 months.
I remember turning to my parents and saying, I'm really scared. I mean, do you think I could die before they figure out what it is?
Finally, doctors discovered the source of her distress: E. coli bacteria in the spinach. Bush's experience is not uncommon. According to the Centers for Disease Control and Prevention, around 48 million Americans get sick and 3,000 die from food poisoning every year. The new genetic technique is helping scientists connect the dots in seemingly unrelated outbreaks of foodborne illnesses. Comparing whole genome sequencing on DNA samples from different people who've become ill allows them to see if the bacteria are the same strain and how resistant they are to current medicine. Initially, health workers sent samples to the CDC, but now all 50 states are getting money to help them set up their own labs. It speeds up the analysis.
It takes about 40 hours to sequence, and you probably have 8 hours of prep time before going on the instrument. And then once you have the sequence information off the sequencer, it's kind of like having a shredded paper document. So we have to use computational biology techniques to piece the genome back together, and that'll usually take a few hours. So all in all, the process from start to finish can be achieved in less than 72 hours.
While initially focused on listeria, the technique was effective for identifying E. coli strains.
So for the current romaine outbreak, we have been able to get some sequencing results back. We can see that the bacteria in that outbreak are very closely related to one another.
By the end of the year, labs expect to use this technique for other common causes of food poisoning. For writer Faiza Elmasry, I'm Faith Lapidus, VOA News.